Now we know that the virus is not a cell, but an infectious agent. Then, how does this agent multiplies? It depends mainly on host cell, and we will understand this fact in this section. Viruses differ in their structure and genomes, which have a very important implication for the mechanism a virus uses to multiply within the host cell. Despite these differences, a general pattern of the viral life cycle is described in the figure you see. These steps are similar, but you need to know that the most important step for a viral infection is to attach to the host cell. And we already know that a virus is host specific. So, the first step in this life cycle that you see in the figure is attachment to the host cell, or a process called adsorption for viruses. This process is mediated by an interaction between receptors on the surface of the host cell and molecules on the surface of the virus. Don't you think that this is amazing? How this process begins and all the steps that are involved. An acellular agent is able to overtake the cell and control it. Think of it. The attachment of the virus to the host cell is followed by entry of either the nucleic acid or the viral nucleic acid into the host cell cytoplasm. If the nucleic acid enters, uncoating of the genome usually occurs before the life cycle continues. A virus can enter the host cell by 3 different modes. The first one is fusion of the viral envelope with the host cell membrane. This nucleic acid will enter. The second one, entry by endocytosis in vesicle. That is, the whole virus will be engulfed inside the host cell. The third one, the simplest one, is simply by injecting the nucleic acid of the virus to the host cytoplasm, leaving the capsid outside. Okay, now the virus attaches to the host cell, and then the nucleic acid is injected into the host cytoplasm. The synthesis of the viral particle begins. During this stage, the genes of the virus are expressed, but how? This when the virus overtake and control the host cell, forcing it to manufacture the viral genome and viral proteins. After the viral particles are produced, which are the proteins and the nucleic acids, now the assembly stage started. The nucleic acids are constructed by self-assembly, which means that this process of putting together the nucleic acid and proteins is coded inside the viral proteins without the need of the host cell to participate. The last step in the viral life cycle is the release from the host cell. Now, mature viruses escape the host cell and going to the adjacent cell to infect them. We learned from this topic that a viral life cycle comprises of 5 major stages. The most important step is the attachment of the virus to the host cell. After the virus assembles, the virus has to escape the host cell to infect adjacent cells.